is Michael Morrison. I'm kind of going to go back to what Dr. DeLucas was kind of talking about at lunch with doing experiments that we can kind of do on the ground, but in a low orbit environment to kind of see how this microgravity environment affects what we're doing. However, instead of doing protein crystals, I'm actually looking at how bacteria grow and adapt in this unique environment that can't be found on Earth. And so just some background on how life adapts to space. Probably the best studied organism that we know of for space are humans. We've had a continuous presence in space for the last 17 and a half years. And that's just if you consider the International Space Station. Before that, there was a space station Mir, Skylab, Apollo, Vostok. And so we've had about 60 years of studying how humans adapt to spaceflight. And what we found, as he pointed out, was that astronauts lose bone density and muscle mass in space, but they also experience this fluid redistribution because we've evolved with gravity constantly pulling down our bodies. So we have mechanisms in our bodies to help push this fluid back up, to kind of keep our blood circulating, and everything kind of working in a nice harmonious manner. And this dysregulation when you are of fluids, when you get into space, because gravity is no longer pulling down, you can stress the body out, which can cause dysregulation of the immune system, which has been seen in astronauts coming back from space as flare-ups of different dormant viruses, such as Epstein-Barr and herpes. And these astronauts we send up there are, very active, healthy individuals that we send up there. For example, right, this photo right here is of, oops, oh, my bad. This kind of, is there a click? Okay, well, where's there a little? The top button, okay. So this is astronaut Tim Peake. In this photo, he was competing in the London Marathon, where he actually set the record for the fastest off-world marathon time at 3 hours and 35 minutes and 12 seconds. And so if we're, seeing these effects in astronauts who are really physically fit, what would we see in humans, such as myself, who a marathon would probably kill? So this is kind of an astronaut's kind of idea, but these are macro, big organisms. My focus is more on the prokaryotes because we keep sending humans to space, but prokaryotes go with us everywhere we go. They're essential for our immune system, our digestion. And does it affect these prokaryotes? And it actually does. What we've observed is that prokaryotes have faster growth rates in space, and they can reach final cell density, higher final cell densities. Some experiments also suggest that antibiotic resistance increases in space, as well as virulence. And this is not things you want if the immune system becomes dysregulated. It's a big concern for NASA for sending astronauts past low Earth orbit, because if you send them out there, they're halfway to Mars, they get sick, you can't bring them back. So we have to figure this out kind of thing. And last, what we kind of notice with prokaryotes is this alteration in biofilm formation. Biofilms do have some role in chronic infections and antibiotic resistance, but NASA's noticed that they affect the systems on the space station as well, such as the space station Mir. It actually clogged the water reclaiming system with a biofilm, and they had to completely rechange out the entire system. And also they find biofilms on the surface of the capsules, which can actually corrode the material, and that's not something you want. Again, halfway to space, all of a sudden your machine is starting to corrode. What are you going to do about it? So this is something that NASA's very interested in. It's kind of what my lab wants to look into, kind of figuring out what's going on here in some kind of molecular responses we can kind of predict and kind of treat. So as Lucas kind of pointed out, spaceflight hardware is important. So for us, we use the biological research in canisters or BRIC spaceflight hardware. It contains a petri dish that we deposited Bacillus subtilis spores, which are a soil bacteria that are non-pathogenic, no risk to astronauts whatsoever. And the spores are dormant, and so when we send them up, we can inject media into them. They become, viable growing cells, and so we can get an effect of what they do just in space without having to worry about the launch and the return. And down here, there's an actual chamber for the media so that it can be stored in until we're ready to inoculate the petri dish. And we used a rich media, TSY, that has protein and glucose as carbon sources, as well as about 10% glycerol, so we could actually freeze our samples and bring them back to Earth, and they would still be in this kind of state of, hey, we're still in space, even though they're on the ground, right when we thaw them. And so the actual timeline for our mission, which became known as BRIC 21, because it was the 21st BRIC mission, launched in April 2015, docked with the International Space Station. They inoculated the samples in space right here. And They were allowed to grow at ambient space station temperature, approximately 23 degrees Celsius, for about 25 hours, after which they were frozen at minus 80 degrees and shipped back to Earth, where me and my lab were able to get our hands on them and start actually processing the samples. And because space is limited, we wanted to do a lot with a little. And so this is kind of everything we wanted to do with them. And you can kind of see we've kind of looked at the antibiotic profiles and the mutation rates and everything. I'm going to focus mostly on the transcriptome, how they're actually adapting today. But I guess it's a little late. But Josh Lehan, a student in our lab, was actually giving a poster on this yesterday. Pretty sure if you still are interested in that, Josh Lehan would be happy to talk to you about mutation frequencies in space. So there's my little plug for him. But from here on, I'm mostly going to be talking about the transcriptome and how the gene expression is affected by space. So how do we do transcriptomics? We get the samples back, they're frozen, we thaw them in the lab, and we immediately extract the RNA. So we have all the RNA that was up in space before it's degraded, as well as the ground samples that we flew, or that we didn't fly, that were sitting at NASA while the spaceflight samples were flying. And we then sequence that RNA to get it, figure out what the actual code is, and then we can do differential expression analysis on those sequences. So differential expression analysis, I take the reads and I filter them for quality, making sure that we actually have good reads. Our reads aren't messed up or anything. And then I align them to the genome, and every single sample we had about 50 million reads per sample. And we had n equals 3 for BRIC21, so that was a total of 6 samples, 3 per condition. And we use a machine to do this because I actually want to graduate eventually and not have to. And then once we align them to the genome, we count how many sequences are there for every single gene, so we can get kind of a quantitative number of how much was this gene being expressed in space compared to on the ground. And then we use statistics. We use actually 2 packages, Lima and DESeq2, to kind of determine if it's higher or lower in space compared to ground. In order for a gene to be considered differentially expressed, it had to have at least a 2-fold change in the number of counts between flight and ground, with a p-value of 0.01. And since we were looking at about 4,400 transcripts, we did correct for multiple testing bias using Benjamini-Hochberg and all that stuff. And so we're pretty confident with our genes we get. And then we do functional analysis to kind of figure out what are those genes actually doing, and do they kind of group into different pathways, so we can kind of try to target pathways, not just individual genes. So we did this on BRIC21, and we found 293 genes that were differentially expressed between flight and ground. So there's about 177 that were higher in spaceflight and about 116 that were higher in the ground samples. And from this, we did our functional annotation, try to figure out what was going on. And we found about 10 enriched KEGG pathways. KEGG pathways are just kind of metabolic pathways such as biotin metabolism, arginine. We actually had a couple non-ribosomal peptide biosynthesis pathways going on, and some nitrogen metabolism, just to kind of name a few. But while we were analyzing this data, NASA was generous enough to let our lab kind of participate on a second spaceflight experiment, which became known as BRIC23, where we flew the exact same hardware, exact same strain, exact same media. And this was very important because to this point, no transcriptome for bacteria has ever been duplicated in space. So this was kind of a nice idea to say, like, can we see the same results if we duplicate this in space? The gold standard of science. And however, during the kind of development and planning stage, instead of growing it for 25 hours, we grew these for 26 hours. That's the only difference between these two. We actually wanted to look at stationary phase, which the first ones were an exponential phase, to kind of see like differences in growth phases. But if there's an effect by spaceflight, we should still see genes up regardless of what growth phase it's in. So can we kind of use these 2 different phases to pull out genes that spaceflight affects continuously? And so these flew about a year after the first ones. And then so we did transcriptome profiling, same way as the first one. And I love this photo because these are actually our samples in space. And they're floating. It's cool. I just love looking at it. There's no strings attached. And so we found about 255 genes in the BRIC23 samples that were differentially expressed. So a few less than BRIC21, but about the same number. And we found about 7 enriched pathways among these 255 genes. And this is all great and everything, but how do they compare to one another? That was the big question. And so I want to know what the variation between the different samples were. Are their samples clustered together? Are they completely all over the map? And so I ran a principal component analysis. And what you can kind of see is if you look at the first principal component, you see that the samples kind of clustered between BRIC21 and BRIC 23, and had about 51% of the variation between all the samples. And because we grew them at different growth phases, this isn't uncommon because they should have completely different gene expressions when they're rapidly growing in exponential phase. That's when they're dormant in stationary phase. But if you look over at the second principal component, you see that about 21% of the variance between all the genes can be explained by grouping the ground samples together and the flight samples together. So we do see this effect of spaceflight even though our samples were going at this different phase, so we can actually pull out maybe some effect of spaceflight even though it's not the dominant source of our variance. And so we then want to see, can we actually see this at the actual gene level? Do we have any genes that are differentially expressed in both experiments? And we found 91 genes that were differentially expressed in both experiments, so yay. And so 55 of these genes were upregulated in spaceflight, which include our biotin metabolism and biotin uptake genes, as well as our biofilm biosynthesis genes. As I mentioned before, NASA is very interested in these biofilm biosynthesis genes. And then we found 36 that were upregulated in the ground controls, and these were nitrogen metabolism in 2-component systems. Nitrogen metabolism is a bit of a misleading because it was really just the nitrate reductase genes, which are expressed in low oxygen conditions. And this is, we also saw some other fermentation genes that are also expressed in low oxygen conditions. So we think most of the genes, it looks like most of the genes that are downregulated were due to maybe a difference in oxygen between the flight and ground samples. But the flight samples, going back to those, we did see this biofilm biosynthesis gene, which we're kind of like wanting to dive in a bit more into, like, okay, we see them up, NASA's kind of interested in these, what do we actually see with our gene counts? And I know probably most of you do not understand what these 4-letter abbreviations are, so I will kind of dive into that. All these genes are the genes required to make biofilms in Bacillus subtilis, the organism that we're using. So if any of y'all that aren't familiar with biofilms, the most simple and probably one you associate with the most is plaque on your teeth. It's a community of cells that kind of group together in a protein-sugar matrix that kind of share nutrients, kind of help each other out, and this protects them from invaders, antibiotics, and a bunch of other environmental stresses. So, bacteria love to grow in these biofilms. And they're made up of, like I said, exopolysaccharide, or EPS, sugars. There's a biofilm surface layer protein, and then we also have a protein scaffold that kind of holds them together as well within the sugar matrix. So we have all of our genes here. And if you look at, this cutoff of it, this is a log 2 scale, so this is a 2-fold change. There's only 2 genes that don't have a 2-fold change between flight and ground. So almost all of our biofilm biosynthesis genes are upregulated in spaceflight. And so this is the first molecular evidence that we have that points towards spaceflight, promoting biofilm biosynthesis. Now, we don't have a reason why this is right now, but something that we're hoping to look into in the future with more spaceflight missions, and more tests on regulations. So just kind of with that in conclusion, we did find about 91 genes that were differentially expressed, and these 91 genes we're going to look further into because these could be good candidates for how space is affecting bacteria. Like, what are these, if we knock out these 91 genes, one of them, how does it affect spaceflight? Does it have an effect at all? Kind of dive in further. Also, look further into biofilm biosynthesis. We do see the first molecular kind of evidence for this increased production in space. And it appears that, at least for the hardware we're using, the availability of nitrogen, oxygen is different between flight and ground, and that's something that we are discussing with NASA on kind of making adjustments to the hardware for that. But, science isn't a one-man show, so I have a lot of people I want to thank. Primarily my boss and Patricia Fajardo, who actually helped with a lot of the processing and de-integration of the spaceflight experiments. The BRIC 21 and BRIC 23 teams, who helped integration with the preparing of the samples and everything, and the astronauts who actually ran my experiments in space that they wouldn't let me do. And so with that, and NASA for funding me, and and with that, I'll take questions.
Thank you, Michael. Any questions?
Do you have ideas about why biotin is upregulated? Is it a general stress response? Do you think there's a certain protein that is a cofactor that's hindered in some way? Like,
So yeah, we're confused about that a little bit. We don't know for sure because it's a B vitamin. It's used in the biosynthesis of some fatty acids, some branched chain amino acids, and glyconeogenesis. But we looked at all the genes involved in those pathways, and none of them are differentially expressed. So we have no clue why they're up.
Regarding the biofilm production, do you think that it might have something to do with quorum sensing, signal regulation related to motility and zero-G?
So there is some thought about motility in space. We didn't see any motility genes upregulated, but we did see a quorum sensing gene, surfactant, that was upregulated in space. So that probably is one reason why we're seeing the increase in biofilm formation, but we don't necessarily know, why that quorum sensing gene would be higher in space. But we did see one quorum sensing gene.
Okay, let's thank Michael again. I don't think we have.
Okay, oh my bad. I can talk to you after.